Well, it's 1 o'clock. I want to welcome everybody to this session. It's entitled What's on the Horizon in Thyroid Cancer Research. Our presenter today is Dr. Al Copland. He's a biologist at the Mayo Clinic in Jacksonville, Florida. Dr. Copland leads the Cancer Biology and Translational Research Laboratory. His research is aimed at better understanding the mechanisms and pathways of carcinogenesis and tumor progression in order to develop novel synergistic cancer therapies. He is focused on cancer, individualized medicine, and new drug development. So welcome, and Dr. Copland, I will hand it to you.
Thank you, Roberta. I'm Al Copland, and as Roberta said, I'm in the cancer biology department down at Mayo Clinic, and I'd like to talk about some new therapeutic strategies in thyroid cancer, some that have actually made it to the clinic. So my talk background today is first to develop some basic cancer concepts for you, that is cells, genes, and mutations. And you guys may know it well or may not, but I've found that this is often helpful in understanding individualized medicine, and developing new therapies and kind of understanding how these therapies work and how they came about. And so I will talk about this concept of individualized medicines in the sense that patients' tumor gene mutations can guide therapeutic strategies, and novel combination molecular targeted therapies are leading to enhanced patient survival. And also discuss a couple of immune-based therapies that are actually in clinical trials, CAR T therapy, ICAM, one, and there's also a novel medullary thyroid cancer CAR T therapy in clinical trials now. And I'll talk a little bit about neoantigen vaccine. We're doing some here, and it may or may not be an effective strategy, but it certainly is interesting, rebooting or developing neoantigens specific to the tumor and vaccinating a patient based on those neoantigens that were discovered in the patient's tumor. So I'd like to start off and give you some background. As you all know, we all start off as one cell, and what you see in the left-hand panel here is a fertilized egg, and in the center of this fertilized egg you see the mother and the father's genetic material. This genetic material is what makes us who we are. And you know also within 9 months, we become a human being from this one single cell. So we're made up of about 50 to 100 trillion cells. That's a 1 with 14 zeros behind it. And these cells dictate our health and the disease process of our bodies. And so any one of these cells can become unhealthy and lead to a disease process in our body. So really, if you think about it, you know, our lives truly are a miracle, and every day is truly a miracle for us. And I think we all know that we have 23 pairs of chromosomes that make up humans, or a total of 46 chromosomes. These chromosomes are made of 3 billion DNA bases, and these in each of our diploid cells. Okay, and for those of you who are a little savvy, you can look at in the left-hand panel here, and you can figure out if this is represents a male or female chromosome. And in the cartoon to the right, you can also tell as well. Males are XY and females are XX. And for the astute, and you'll probably understand why females are smarter than men. Men are missing a lot of their DNA in their 23rd chromosome, the Y chromosome. So these DNA base pairs are pretty simple. guanine, cytosine pair with one another, adenine and thymine pair with one another through these hydrogen bonding processes. And essentially, they make a double helix, or even kind of like a slinky, and making a double slinky, and make this double helix that can actually, just like a slinky, you can pull apart. And when it's pulled apart, these genes are transcribed, or you can keep it very, very compact, and the DNA is quiescent or little is going on transcriptionally in that part of the DNA. And here you can see in the bottom right-hand corner the A's and T's bonding together, as well as the C's and G's, and this winding staircase of this double helix. So nature's been incredible. And so here you can see in the left-hand panel the double helix. So when cells replicate, they have to replicate all of the DNA, all of the chromosomes. And so this DNA actually unwinds and actually replicates itself. And so you can see the light gray is the template, and the pink is actually extending and adding these bases. And it's the sequences of bases that dictate all of the phenotype and who we are. And so these mistakes can be made here and add incorrect DNA bases or mutations can occur at this particular point. And so this might be a step where cancer can be potentially initiated. This is a beautiful picture of cell division. And so you can see at the very top at interphase, where the cell is starting to divide and separate its chromosomes all the way through the steps of prophase, metaphase, anaphase, and telophase. And at telophase, you can see where the cell is actually dividing into two cells. And so essentially, cancer is a genetic disease, and it's mutations, and in solid tumors, it's multiple mutations that have to occur for a tumor, a normal cell, to become a tumor cell. And so here you've got a cell with all its complexities. This towards the center of the cell is the nucleus. This is where the chromosome is housed. It's also housed in mitochondria as well. But you've got this powerhouse in the nucleus. Another complexity is that you've got your genes in your DNA, and from this DNA, genes code for proteins, and it goes through this process called transcription. So it does this replicative process where messenger RNA is coded similar to replicating DNA off of the DNA template. These messenger RNAs are then translated. 3 mRNA bases code for very specific proteins, or peptides or amino acids, and these amino acids are translated into proteins with very specific amino acid sequences. So we think there are around 150,000 proteins that can be translated off of about 22,000 messenger RNAs with different splicing events. So you can begin to see how complicated our human body is, and steps where mistakes can be made. So mistakes can be made in replicating the DNA itself. Steps can be made in transcription of the messenger RNA. Mistakes can be made in the translation of these proteins, that we get mutated proteins that can become oncogenic, causing cancer, or proteins that are tumor suppressor genes that stop cancer from forming. Another important concept to know is that 95% of all cancers are sporadic. That is, our genome is perfectly normal and fine. It's how we interact with the environment and damage our DNA that causes cancer. There's only a very few inheritable gene mutations, about anywhere from 2 to 5%, that you actually inherit from your parents, and those mutations will be in every cell of your body. And those mutations typically occur early in life, and so you get a number of hereditary childhood cancers that are well known and characterized and very hard to cure. So the reality is, it's thought that up to 50% of cancers are potentially preventable. Some of the environmental causes of cancer you all well know: tobacco, diet, obesity, sedentary lifestyle, different occupational environment hazards such as asbestos, coal dust. We have numerous viruses and infectious agents that can induce cancer. We well known as HPV, alcohol consumption, ionizing UV radiation such as the sun, and a small percentage, as I said, are inherited gene mutations. So we do have the ability to potentially prevent cancer in our own lives based on how we take care of ourselves. So again, this idea of environmental risk factors that can target your genes, cause mutations, and multiple mutations will lead to cells that essentially have the ability to be, the more mutations you get here, you can see you get precancerous lesions, cancer in situ, is that in the organ of origin, such as the thyroid, and then you can have invasive and metastatic cancer that metastasizes to different parts of your body. And so this idea that we have cancer cells lose this sense of homeostasis, that is, this balance of cell birth and cell death. Cancer cells are essentially immortal. That is, they replicate and they continue to replicate, and they don't lose that ability to replicate and die. And essentially, our normal cells have a certain number of times that they're going to replicate, and this is part of our aging process and our own mortality. Importantly, you may not think about it, but about every second, there are about 25,000 cells dividing in your body. And you know that we're not getting any larger as adults, so there's a homeostatic process. If there are 25,000 cells dividing, then there are another 25,000 cells that are dying off. So always like to say is, some days you wake up and you think you feel like a new person. You just might be, because of this process. So at this point of the talk, there should be some take-home facts for you. We're made up of cells, and it's about 50 to 100 trillion cells. Each cell contains about 3 billion DNA bases that make up our chromosomes, or essentially our identity. Our cells replicate in our body over our lifetime, duplicating DNA, and this occurs often and constantly. So you think about hair, in other parts of your body that are growing all the time. Each replicating cell in our body also has the potential to become a cancer cell. Most mistakes in DNA replication are translated to proteins that then cause cancer, as well as proteins that are silenced that are known to prevent cancer, or tumor suppressor genes. So cancer cells are programmed to replicate forever, that is, they're immortal, and have the ability to metastasize and take over other organs, shutting down that organ and causing the death of a patient. So I think hopefully that these basic concepts give you an idea with regards to understanding cancer and cancer cells and the complexity and the miracle that we are. There are known hallmarks of cancer, and every cancer cell has these hallmarks, and they're genes that have been altered or mutated that allow the cancer cell to have these phenotypic properties. So there are genes that regulate proliferation, that is, these cells are replicating continuously. And if you think about it, if one cell replicates every 24 hours, just do the math, double a cell every 24 hours in 30 days and look at the number that you come up with. It'll be astounding. It's around 50 million. So take one cancer cell, replicate it every day. So 1 becomes 2, becomes 4, becomes 8, becomes 16, becomes 32, becomes 64, and on. In 30 days, if one cancer cell replicates, you've created essentially a monster within your body, especially if it has the ability to metastasize. Also, cancer cells have the ability to evade growth, suppression, and so they have the ability to continue to proliferate as well as evade suppressive mechanisms. Our normal cells, when they contact one another, stop growing. That is, they don't replicate anymore. Whereas cancer cells don't have that signal. They've lost that signal. So cancer cells will continue to replicate and just stack up on one another. Also, cancer cells have the ability to avoid the immune system and not be destroyed by the immune system by turning on certain genes that block the ability of immune cells to attack the cancer cell. And we're learning quite a bit of that in the last 5 to 10 years and now have therapeutic strategies. They, as I told you, they're immortal. They don't die. And so they can continue to replicate forever. And you all probably know the story, the HeLa story of the first cell line that was ever developed in the 1950s. We still use those cells today. Also, tumors promote inflammation. This inflammatory process affects all of these other processes, especially immune evasion. And importantly, you know that cells can, tumor cells activate invasion and metastasis. 90 to 95% of all patients die from their metastatic disease, not from localized disease. Inducing angiogenesis. All cells need blood vessel supply, and after about 2 to 5 cubic millimeters, cells have to be able to make their own blood vessels. And so cancer cells have the ability to do that. This unstable cancer genome allows cancer cells to continually to mutate and become more aggressive and deadly over time. And of course, we know that cancer cells can have lots of different mechanisms to resist cell death. And again, cancer cells can deregulate cellular energetics. We typically use glucose as an energy source. And I'll tell you about a pathway that we've discovered or that is well known, utilizing fatty acids, and we've developed a small molecule inhibitor to block that signaling pathway. And so here you see it looks complicated, but again, just to give you an illustration, here's the EGF receptor. It's a tyrosine kinase membrane protein that sits on the cell surface. So this line, this little dark line, is indicative of the cell membrane, and it signals through multiple protein signaling pathways to regulate cell survival as well as promote cell proliferation. And again, these genes under normal circumstances are critical to our survival, but also when deregulated, we know that the EGF receptor can be mutated and turned on all the time. We know the RAS gene can be mutated as well in thyroid cancer, PI3 kinase, AKT. So if you guys are familiar with any of the molecular or mutations in thyroid cancers, these are well-known pathways in thyroid cancers that are dysregulated. And so, and we now have therapeutic strategies to target actually each one of these proteins now. And so what I would like to propose to you is that what's not been done is testing combination therapies targeting some of these newer therapeutics. So we now have a new therapy against RAS, new therapy against PI3 kinase. You know that we've got MEK inhibitors and RAF inhibitors for RAS and BRAF mutated tumors that provide survival benefits for patients. So these are well-known mutations that occur in thyroid cancer, starting with the thyroid follicle. And you know, very common, the most common form is papillary carcinoma, followed by follicular carcinoma. And you know that these are differentiated thyroid cancers that are easily treated with radioiodide or surgery if they're still localized. But they can de-differentiate to poorly differentiated cancer and to completely de-differentiated anaplastic thyroid cancer, which is the deadliest cancer known to humans. And we know a number of the mutations that are driving this. So RET-PTC drives papillary carcinoma. We now have molecular targeted therapies that target the RET protein. So cabozantinib, bandetinib, selpercatinib, which is the newest RET inhibitor that's just been FDA approved, that's very specific for RET, whereas these others, lenvatinib, sorafenib, are tyrosine kinase inhibitors, but they inhibit other tyrosine kinase receptors as well. And so we're now developing these molecular targeted therapies to really target known mutations. Just recently, sort of received is a very specific KRAS small molecule inhibitor of KRAS, and it's been FDA approved in non-small cell lung cancer. We know that RAS is mutated very often in papillary cancers and anaplastic cancers. Regretfully, KRAS is the least often mutated, with N and H being the most commonly mutated found. And in follicular adenomas that go on to follicular carcinomas, we know the RAS PI3 kinase pathway, oncogenic pathway, P10 is a tumor suppressor gene, and you've got the PAX8 PPAR gamma that are known to cause follicular carcinomas. We also know that TP53 is a very often mutated tumor suppressor gene. We now have a small molecule in reactivator, so it reactivates P53 if it's still being expressed, mutated P53. And so this is actually in clinical trials right now. We also have an inhibitor to PI3 kinase called lipolysis, which is FDA approved as well, but they've not been tested in thyroid cancers as of yet. And so these are really new therapeutic strategies that can be tested in preclinical models that might demonstrate clear anti-tumor synergy when combined in patients with these known mutations. And you all do know that dabrafenib plus a MEK inhibitor, trametinib, are FDA approved for anaplastic carcinoma and showing survival benefit and prolonging lives of patients diagnosed with anaplastic thyroid cancer. And down at the bottom is just for anaplastic thyroid cancer, the percent mutations that are seen in anaplastic thyroid cancer. So you can envision potentially putting different combinations of these new therapeutics together to come up with novel combination therapies that might provide survival benefit and might even lead to cures later down the road. So these are things that are yet to be done, and I'm very excited about this concept of combining molecular targeted therapies to enhance survival benefit and quality of life. Just to give you a little bit more in-depth idea of what each RAS signaling pathways do, which is a very commonly mutated pathway in thyroid cancer. You can see that HRAS is critical under normal situation, important to cell proliferation and cell migration, and it's activated by these receptor tyrosine kinases like the EGF receptor that sits on the cell membrane. And HRAS gets activated. It then activates PI3 kinase. PI3 kinase then activates AKT, then activates mTOR, which is critical to cell migration, which can lead to metastatic metastasis of tumors to other organs. You can also see on the right-hand side that HRAS signals to the RAF. There are 3 RAFs, A, B, and C RAF, and the RAFs then signal through MEK, then on to ERK, and then ERK can activate c-MYC, a transcriptional factor that's oncogenic when it's overexpressed, and it can drive this epithelial to mesenchymal transition, which again causes these tumor cells to be more migratory and invasive and metastatic. And so you can see how oncogenic RAS can drive cell proliferation, cell migration, and invasion, as well as enhanced protein synthesis through signaling through mTOR. And so understanding these molecular signaling pathways really helps us to start developing effective combination therapies and understanding how these pathways are working. And resistance does occur through these pathways, and we have to understand those mechanisms as well. This is some work from Rebecca Schweppe's lab, and Rebecca discovered Sarc inhibitors, dasatinib, which is FDA approved, has anti-tumor activity in aggressive thyroid cancers. And what she also found was that these tumor cells develop resistance. And she discovered that the resistance is through when Sarc is inhibited, this other protein called ThyCa is upregulated. And so here on the right-hand side of the panel here, you can see if you inhibit Sarc, it actually upregulates ThyCa. In fact, activates a transcription factor that then upregulates signaling pathways that are important in metastasis, these MMPs, as well as interleukin-1 beta, which is an inflammatory cytokine. And so you can see if one were to block ThyCa, and that's what she did experimentally, and showed now that the Sarc inhibitor does not become drug resistant. So she combined a ThyCa inhibitor, this PF562-271, with dasatinib, and showed that she can reclaim the anti-tumor activity and actually enhance anti-tumor activity. And so in this figure C, you can see the black line essentially is inhibition of growth. So 100% growth is cells replicating and growing and not being inhibited over probably a 3 to 5 day period that these cells replicate rapidly. And so you can see when you combine these two inhibitors, you get enhanced activity at lower concentrations. So this PF562 at lower concentrations. So you can see on the x-axis 0 to 5 micromolar. So you can see 50% growth inhibition at 0.312 micromolar. Whereas you don't see 50% inhibition with the single agents until out at 2 to 5 micromolar doses. So you can see that combining these two compound and drug, you sensitize the tumor to growth inhibition and cell death. And so again, these are strategies that can really enhance tumor death and patient survival. This is another nice article, a review article from Rebecca, just trying to take these concepts home more. Again, the receptor tyrosine kinases that sit on the cell surface with the EGF receptor and others, and all of these new drugs that we now have that we can begin combining and testing for anti-tumor synergy. And so you can see all of these pathways are critical in thyroid cancer that we can now test in combination to hopefully develop. Yes.
Dr. Copland, we have a quick question.
Yes.
Well, there's a couple. What does oncogenic mean?
Oncogenic means that it causes cancer. So there are genes that can be put into a normal cell and turn that cell into a cancer cell. And so RAS is an oncogene. RAF, so mutated RAS that is turned on and is making active RAS protein all the time is an oncogene. RAF is an oncogene as well. PI3 kinase and AKT are oncogenic as well. Great question.
There's another one. Why do some cancers grow slowly and others more rapidly?
Great question. Exactly the same thing. You've got these oncogenes that are driving cell proliferation. And so the more oncogenic activity you have, the faster these cells will replicate. And so cancer cells can replicate as fast as we've got some cell lines that replicate in 15 hours. It's scary. And other cell lines might replicate at 55 hours or 80 hours. So they're slow growing tumors. And it has everything to do with the oncogenes that have been mutated and the level of their mutation, or the combination of oncogenes that are promoting the ability of these cells to completely replicate themselves in a shorter time frame. Does that make sense?
Mm-hmm. There's one other one, and we'll have you move on.
Okay.
What does it mean when suppression therapy stops working?
You become drug resistant. So you can, and the example that I just gave was this mechanism that Rebecca discovered with a Sarc inhibitor. She was showing repression with a Sarc inhibitor, and over time was losing that repression, and was losing that repression because Sarc was actually upregulating this gene or protein called FAC. And this FAC protein promotes cancer growth. And so you can lose drug sensitivity through multiple mechanisms. And this is also especially true of the tyrosine kinase inhibitors, where other pathways are upregulated. When you block one cancer-causing pathway, you can upregulate another cancer-causing pathway. And these are how cancer cells are protecting themselves and surviving. And so that's why it's so critical for us to study these molecular mechanisms and signaling pathways, understand how the cancer cell is mutating or altering its ability to survive through other signaling pathways. So there's lots of redundancy in different oncogenes that can drive cell proliferation and cell survival and drug resistance. And so it's just not one single pathway. There are probably 40 or 50, 100 in different combinations and permutations. And that's why cancer is so complicated. It's not one single gene that's been altered. It's many, many genes, and you can block one and then flips on another. So it's wonderfully intriguing, but at the same time kills patients because we have not figured out these combinations that need to be figured out.
That's it for now.
Okay. Please, yes, thank you for the interruption, and anytime, just stop me. This is actually what we call a heat map, and we've done genomic profiling on patient tumors. And so these are matched samples of normal tumor, normal tissue, normal thyroid on the right-hand side. So NT1 through NT13 are normal thyroid tissues where we've isolated RNA and done whole gene sequencing of the 22,000 genes that are in each cell. And you can see we've also done RNA sequencing on 12 patients. And through this analysis, we can determine which genes have are commonly altered, upregulated, or downregulated that might play a role in the tumor biology. And one gene that we discovered is called SCD1, or steroyl-CoA desaturase 1. It's the rate-limiting enzyme that regulates the synthesis of monounsaturated fatty acids, which can be used as another energy source as opposed to glucose. And so you can see in the anaplastic tumors, SCD1 in red being hot or upregulated, is upregulated in every tumor that we looked at. And you can see blue, cool or lower expression levels in normal tissues, in all normal tissues. So this is a gene that is commonly overexpressed in deadly anaplastic thyroid cancer. And so we went to explore whether we could block the expression of this gene and if it made a difference in tumor growth. We first looked at the expression of SCD1 in patient samples, and you can see that it is expressed in normal tissue. You see the light brown staining, but there's little to no staining in the normal cells. You can see the ring and the blue nuclei that show little to no staining in normal cells. You get to follicular adenomas, you can start to see not really, again, you look at the blue nuclei, there's really no staining around those blue nuclei. And so there's little to no staining in normal tissues. Now you get to papillary PTC, non-invasive versus invasive, and now you can start to see very strong brown staining, which says that the protein is being expressed. And you can see, we can quantitate this, we call these H scores. And you can see this H score jumping up to 169 in these invasive papillary thyroid carcinoma tumor tissues of patients. And looking in follicular thyroid carcinoma, you can see non-invasive, there's no staining of SCD, and very strong staining in SCD. And then in anaplastic, again, you can see very strong staining of steroyl-CoA desaturase 1 in anaplastic thyroid patients. And so we blocked this signaling pathway, and Christina von Rumling, who is a technician in that, she's got a doctorate degree, and she's Dr. von Rumling, worked on this project a few years back. And so let me just tell you a little bit about SCD. It's the rate-limiting enzyme that converts saturated fatty acids to monounsaturated fatty acids. And like I told you, this pathway is highly upregulated in aggressive tumors. And it's very important because it's an alternative energy source for rapidly dividing cancer cells. So they need to upregulate this pathway. And they do it through this transcriptional factor called SREBP1, which regulates all of the enzymes involved in making these monounsaturated fatty acids like oleic acid and palmitoleic acid. Also, these fatty acids are very important in integrating into cell membrane structure. And so, again, this is a pathway that is upregulated in rapidly dividing tumor cells. And it's a pathway that is common across cancers. Here you can see a laundry list of tumors, and I've just highlighted thyroid cancer, and you can see it's highly upregulated in thyroid cancer, as well as many other tumor types. So this is a pathway that is commonly activated in aggressive tumors. And So we have a talented assistant professor here, Tom Caulfield. Tom does in silico drug model. So we can now have the ability to design drugs on computers. So we can create three-dimensional models of these proteins and identify binding sites. And so in the right-hand corner here, you can see a binding pocket, and we can dock and develop any molecule that we want and dock it in this site to see how it fits. And the better the fit, the more likely that it will block the ability of this protein to be biologically active. Does that make sense? So we have technologies now that we can better predict and develop new drugs. So using this technology, we developed brand new compounds that have never been synthesized in the world before. So we had 40 of these synthesized, and of those 40, 4 of those compounds actually are SCD1 inhibitors. This is our lead compound that we call SSI4. And this small molecule, we know that it can be taken orally. It has very good oral bioavailability. It's very well tolerated. It is very specific for targeting SCD. It does not have off-target effects. And it has a very high binding affinity for SCD1 with an IC50 of 1.9 nanomolar. And it has broad activity against many cancer types, including, as you saw, anaplastic and papillary thyroid cancers, or as you're about to see, I'm sorry. So these are cell lines that we have screened. Importantly, it has no effect in normal cells. So we can culture normal thyroid cells. And here's an example of 3 in the top left-hand corner. And you can see over a 1 nanomolar to 10,000 nanomolar or 10 micromolar dose of an SCD1 inhibitor, there is no effect on cell proliferation. That's also true for follicular adenomas. You can see no effect. So normal cells are not affected by blocking SCD. Here you can see in the bottom left-hand corner in papillary thyroid cancer cell lines, you can start to see a number of these cell lines become responsive. These more aggressive cell lines are responsive and growth inhibited. And in anaplastic in the bottom right-hand corner, you can see all 3 of the cell lines tested here are highly sensitive, and they're sensitive in 1 to 10 nanomolar range with IC50s of 1 to 10 nanomolar. So you can see that blocking this pathway is a critical survival pathway, and that these aggressive tumor cells are dependent on these monounsaturated fatty acids, oleic and palmitoleic acid, that are highly abundant when this pathway is upregulated and turned on. We also show that blocking SCD1 in combination with other drugs is highly synergistic. And again, here's a growth curve, and you can see when SCD1 inhibitor plus a proteasome inhibitor are combined, you can see inhibition at lower concentrations, much greater inhibition with these drug combinations. Here's an example of in vivo, and this is a combination of sorafenib, which is a tyrosine kinase inhibitor that you saw earlier that blocks the RET pathway, in combination with our SSI4 compound, and you can see strong combination synergy with little tumor growth in the purple, and you can see the single agent activity of SSI4 in the red and sorafenib in the green, and the control vehicle placebo, where tumors grow very, very large, very rapidly in these animal models. This is the lab and collaborators within the lab, and Dr. Bob Smallridge, who's been a dear friend and mentor, who's one of the world's experts in thyroid cancer. And many of you may know Bob. I'd like to end on talking about 2 immune-based therapies, CAR T cell therapy and neoantigen vaccination, which is pretty new on the horizon. And the exciting thing is there are actually 2 CAR T clinical trials ongoing in thyroid cancers right now. So what is CAR T therapy? You basically take a patient's immune T cells, remove them either through a bone marrow biopsy or activating T cells to be mobilized out of the bone marrow into the blood and collecting T cells. Those T cells are then grown in cell culture and they can replicate into millions of cells. These cells are then engineered with these chimeric antigen receptors, or these CARs. And so basically these CARs are inserted into all of these T cells, and then these CAR T cells are given back to the patient. And now these CAR T cells are armed to go attack the tumor and actually engulf and destroy the tumor cells. So these CAR T therapies have been proven to be powerful in hematopoietic blood cancers and curative in many instances. The problem with solid tumors is really identifying a protein that is uniquely expressed on the cell surface of the tumor that can be targeted and not expressed in normal tissues. Because wherever that CAR T cell sees that protein of interest expressed on the cell surface, it will attack it and destroy it. So if it's expressed in a normal organ, it will attack that normal organ. But there's clearly progress here. So we have, like I said, 2 phase 1 clinical trials that are on clinicaltrials.gov. One is in iodine refractory, poorly differentiated thyroid cancer and anaplastic thyroid cancer that's being done at Cornell. And it's the antigen or the protein that is being targeted is ICAM1 that is expressed on anaplastic thyroid cancer as well as refractory. It's also expressed in other cells, but this group is taking advantage of creating a low affinity antibody and trying to tune it in so that it's only effective in high expressing situations like anaplastic or poorly differentiated thyroid cancer. So I really look forward to seeing the results from this trial. Also in medullary thyroid cancer, there is a CAR T that targets this glial cell derived neurotrophic factor receptor alpha 4. And interestingly, this GFR alpha 4 plays an important role in autophosphorylation and activation of the RET receptor. And so both of these trials should be very interesting to follow and to see the outcomes in these trials. I think,
Dr. Copland?
Yes, it's
we have just under 10 minutes.
Yep.
So I wanted to just let you know that we've got a little time and we've got some more questions.
Yes, I will. You want to take some questions? Let's take some questions, and I'll skip this explanation of CAR Ts for sure. So what questions do we have?
Okay, so this one is there research looking at lipolysis in relation to TSH suppression, i.e. does TSH suppression halt lipolysis?
You know, I don't know the answer to that, and I will look it up, and I'm happy to get back with whoever. I do know that there are clinical trials ongoing with targeting TSH. I actually was looking at it earlier this morning, but I will look at the relationship of TSH and lipolysis. Typically, we think of TSH as stimulating cell proliferation through the TSH receptor. And I'm writing myself a note to look at that. Go ahead.
Okay, there's one. Why don't the other pathways you said would become active when the drug acts on a specific pathway not work at the same time as the specific pathway targeted by the drug?
Well, if I understand the question, the blocking one pathway stimulates the activation of another pathway. And so say the Sarc pathway was playing a role in driving cell proliferation, that pathway got blocked. And in blocking the Sarc pathway and trying to kill the tumor cell, then blocking Sarc stimulated FAC upregulation, and then FAC now is taking over from Sarc and promoting tumor cell proliferation. I'm not sure if I answered the question, but by blocking one pathway, you can often through regulate cell signaling regulation pathways, the cancer cell, it may be a direct effect of blocking that pathway, or the cancer cell senses loss of that pathway and actually stimulates other pathways that will promote cell proliferation, cell survival, to stay alive.
What gets unregulated if you turn off SCD?
What happens is blocking SCD induces this ER stress, or endoplasmic reticulum stress pathway. And this is a survival pathway. And so these monounsaturated fatty acids are critical in the endoplasmic reticulum, and blocking SCD1 leads to elevation of ER stress, and the number of proteins are upregulated. And if they're chronically upregulated, it will lead to apoptotic cell death. And that's what we have found and published when we block the SCD1 pathway, is that these cells are dying through this ER-mediated stress pathway. And we're also finding some effects on the immune system, and we've got some data that suggests that combining with immune checkpoint inhibitors is synergistic, and we're actively investigating that pathway as well.
Okay, there's another SCD question. Has SCD been tested with Hürthle cell TC with a P10 deletion?
It has not. And I think we've gotten that cell line. We've not really worked with Hürthle cell or medullary thyroid cancer that much. I have a colleague, Anna Chindris, who's working on Hürthle cell and is targeting some other novel signaling pathways in Hürthle cell right now, but it might be worth us testing our SCD1 inhibitor with some of the other inhibitors that I've shown earlier today. And it's certainly gotten me more, this talk has gotten me more interested in testing other inhibitors. So I'm appreciative for this talk for that reason, and getting to talk to everybody. I'm sad that I don't get to see folks in person.
I have two more questions, but did you want to keep going with your presentation?
Yeah, let me see if I can do it real quick. And so our lab has done genomic profiling and identified TSH receptor as a potential antigen to use as a target for CAR T cell therapy. And in the top right-hand corner, you can see this purple line for thyroid, and this is looking at expression across all of the organs in the body. And you can see that the thyroid expresses high levels of the TSH receptor. And so we have made antibodies that target the monoclonal antibodies that target the TSH receptor, and made TSH receptor targeted CAR Ts. And we have done this in animal models and demonstrate a dose responsive inhibition of tumor growth. And so here you can see where we implanted tumors and then treated with either 11 million in the gray, and you can see complete inhibition of tumor growth over 18 days, or 5 million, you can see suppressed tumor growth compared to the vehicle control of 5 million T cells that have not been armed with the TSH targeting mechanism. And you can see a survival curve here on the right, with the black being 50% survival over a 40-day period, with this 11 million CAR T cells on board. This is the team that's working on this project. A number of clinicians. Saad Kandarian is our CAR T expert. He spent 3 years training with Carl June at University of Pennsylvania. He's designed the CAR Ts, and and the other individuals are clinicians and experts on CAR T or developing CAR Ts towards a clinical trial. The other new thing on the block is vaccines, and essentially we're working with Keith Knutson, who's a vaccinologist. Keith is actually running a number of trials in ovarian and breast cancers with vaccines that he has developed. And we have recently developed in one patient done what's called a neoantigen vaccine. And so what that requires is being able to sequence the patient's RNA and DNA, as well as normal DNA, and identifying tumor-specific antigenic sequences that would potentially activate the immune system. And so essentially you're synthesizing those peptides. We typically, like for the COVID virus, the spike protein was used as the antigen to vaccinate folks to protect us from COVID. Well, in this case, we can identify anywhere from 20 to 30 peptides that we can use as immunogens. And so we have multiple chances of developing multiple immune reactive T cells that can attack the tumor if any of those antigens are immunogenic. And so important technology for this is also to have a bioinformatics pipeline that can predict neoantigen sequences. And Jan Osman in our group has developed a neoantigen bioinformatic pipeline over the last 3 years. And so we're now, we'll be testing this in select patients. We've got the IND and FDA approval to do this in a patient moving forward who's got metastatic disease, to see if we can drive an immune system, increase the immune reactivity. So to finish up, take-home messages: cancer is a multi-genetic disease and not a simple cure. Combined targeted therapies provide enhanced anti-tumor activity, and there are many new treatment options that are on the horizon, some that will lead to increased quality of life and cures. And so I'm very excited and hopeful. It's just a matter of money. We've got the technologies and keeping the bright minds engaged to make these discoveries and to develop these new technologies and therapies that can save lives. So I'd really like to thank each one of you. And as Margaret Mead has said, never believe that a few caring people can't change the world, or indeed, that's all we ever have. And I count each one of you in this group of caring people. So thanks for listening, and thank you for spending your time to gain knowledge and try to defeat these awful cancers and diseases. Thank you.
Okay, we have just a couple minutes, but we have a few more questions I'm hoping we can get through. There was one: what's the sequence of testing for these new inhibitors? Petri dishes, then animal studies, then human clinical trials. How long does that sequence of testing typically take?
That's absolutely correct. The in vitro methods are fairly rapid, within months. The animal studies may take 1 to 2 years, depending, if we can do it. My laboratory actually develops cell lines and these patient-derived xenograft mouse models, so that we can, and we also do mutation analysis, so that we can develop novel combination therapies that we can take into clinical trial. So actually, hematology oncology fellow working on prostate cancer in the laboratory, has been working on this project for a couple of years and has a novel combination therapy for castrate-resistant prostate cancer. And he's already generated the data in the animal models and is hopefully writing a trial within the next year, and hopes to find funding to do the clinical trial. So it can move very fast if the therapeutics are already FDA approved, and in this case, they're, you know, the novel combination that he's identified, they are FDA approved, and so he can move rapidly if he identifies the money. So it depends on if the therapies have to go through FDA approval or not. So like our SCD1 inhibitor, we've been working on this for 10 years now, and you know, and we're going through the process right now of generating all of the toxicology data and a number of other data sets that the FDA needs before it deems any compound to be safe to be put into a human. And so that can take a number of years before you can get what's called investigation of new drug approval from the FDA. And that's the process that, you know, there are lots of ways to fail before you ever get a compound into a patient. And thank goodness we have the FDA. They do a wonderful job of protecting us. And
so that's the end of our time. I'd like to thank Dr. Copland for your time today and all this amazing information that you brought to us. It's been really, really helpful, and I know a lot of people will benefit from it. So thank you.
Thank you, Roberta. Thank you, Candy, for hosting.
Yeah.
You're
Thanks
welcome.
so much.